and Assalamualaikum to everyone. My name is Fatin Amalia. Today, in this video, I'm going to share about translation. What is translation? Translation is the process where codon of mRNA is translated into a sequence of amino acid to produce polypeptide chain. This process of translation occurs in cytoplasm. Translation process involves ribosome, tRNA, and mRNA. Ribosome function to facilitate the attachment of tRNA anticodons and mRNA codons, while tRNA function to carry specific amino acid and transfer amino acid from cytoplasm to ribosome. Next is stages in translation. There are 3 stages in translation. Number 1, initiation. Number 2, elongation. And number 3 is termination. While elongation has other 3 steps. The first stage is initiation. A small ribosomal subunit with initiated tRNA bind to the mRNA. This tRNA carries the amino acid methionine. This tRNA then binds to the start codon AUG, which signals the start of translation. Large ribosomal subunit attached to the mRNA and small ribosomal subunit to complete the translation initiation complex. The figure shows the example of translation initiation complex. The initiated tRNA inhabit the P site of the ribosome and A site is available for the next amino acid tRNA. The second stage is elongation. There are 3 steps in elongation. The first one is codon recognition. The second one is peptide bond formation. The last one is translocation. Elongation 1. Codon recognition. Amino acid tRNA that carries the new amino acid binds to the codon in the A site by complementary base pairing between the tRNA's anticodon and mRNA's codon. Peptidyl tRNA that carries the growing polypeptide occupies the P site. Elongation 2. Peptide bond formation. The growing polypeptide attached to the amino acid on the tRNA at A site. Peptide bond is formed between the amino group of the new amino acid and the carboxyl end of the growing polypeptide. This reaction is catalyzed by peptidyl transferase. Elongation 3. Translocation. The ribosome translocates the tRNA in the A site to the P site. The mRNA moves along with its tRNAs and bring the next codon to be translated to A site. The empty tRNA is moved from P site to E site where it is released. This process is repeated until the polypeptide is complete. Next is the last stage, termination. The ribosome reaches the stop codon on mRNA. Stop codon signals the stop of translation. A protein called release factor binds to the stop codon in the A site, which causes the water molecule to be added into polypeptide chain instead of amino acid. Hydrolysis of the bond between the polypeptide and the tRNA in the P site occur. The polypeptide is released from the ribosome. The 2 ribosomal subunit and other components dissociate. Bye, thank you.